Hello and welcome. Today, we're diving into a really important topic for every medical student, Acridine Orange staining. In microbiology, getting answers fast can literally change a patient's outcome, and that's why Acridine Orange is such a powerful tool in our diagnostic arsenal, especially when the clock is ticking. So, to really get why Acridine Orange is such a big deal, you've got to understand the problem it solves. On one hand, you have your traditional stains, right? Like Graham or Leishman stains. They're awesome. They're the gold standard. They give you a ton of information. But they can be slow, and they take a really skilled eye. On the other hand, you've got fluorescent stains. These guys are built for speed. Think rapid screening, super high sensitivity, a quick way to see if there are any bugs in a sample. And that's exactly where today's star player, Acridine Orange, comes in. All right, here's our game plan for this session. We're going to build this up from the ground floor to get you totally prepped for your exams. We'll kick things off with the basics, then get into the cool science of how it works, walk through the actual procedure, check out its uses, weigh the good and the bad, and wrap it all up with a super high-yield review. Okay, so first things first. What exactly is Acridine Orange, and why is it so crucial in modern labs, especially the really busy ones? So here's what makes Acridine Orange so special. It's not just a regular dye that adds a splash of color. Oh no, it's a fluorescent dye. What does that mean? Well, it absorbs light energy at one specific wavelength, and then, this is the cool part, it spits that energy back out as visible light at a different, longer wavelength. It's like it gets excited by one color and then literally glows in another. And that unique glow is what lets us see microbes that would be completely invisible otherwise. The best way to think about it is like this. You know how you use a highlighter to make important text jump right off the page? Well, Acridine Orange does the exact same thing, but for microorganisms. It makes them pop right out, glowing brightly against a dark background. This makes it an unbelievably sensitive and efficient tool for a quick scan of a sample, especially when you need an answer in minutes, not hours. All right, let's get into the nitty-gritty science. How does this all work on a molecular level? Why does it even stick to microbes? And what's with the different colors we see? The whole secret is a process called intercalation. Sounds fancy, right? All it means is that the Acridine Orange molecule is flat, and it loves nucleic acids, so it literally slips itself, or intercalates, right in between the base pairs of DNA and RNA. It just slides in there. And this close and personal binding completely changes the dye's properties, causing it to glow super brightly when you hit it with the right kind of light. Now this is where it gets really clever. Acridine Orange has this property called metachromasia. Basically, it means the dye can stain different things different colors. So when it slots into that nice, rigid, double-stranded DNA, it grows green. But when it binds to the more flexible, single-stranded RNA, it clumps together and glows a brilliant orange or red. This color coding is the absolute key to its diagnostic power. Okay, this leads us to a classic exam question, so listen up. If microbes have both DNA, which should glow green, and RNA, which should glow orange, why do we almost always see them as bright orange little dots? This is a super high-yield concept, so let's break it down. And here is the answer. Actively growing cells like bacteria and yeast are basically little protein-making factories. And to make all those proteins, they need a ton of ribosomes. And what are ribosomes made of? Ribosomal RNA. There's just so much RNA packed into these cells that the orange glow it produces completely overwhelms the green glow from the cell's single DNA strand. So when you see a bright orange dot on your slide, it's a huge clue that you're looking at a living, growing microorganism. Now, to see all this glowing action, you can't just use your standard microscope from the lab. Nope, you need a specialized piece of kit, a fluorescence microscope. This setup has a super powerful light source, like a mercury or xenon lamp. But the real magic is in the filters. First, an exciter filter lets through only the specific wavelength of light needed to get the dye excited. Then, after the sample glows, a barrier filter blocks all that original light, so the only thing that reaches your eye is the brand new fluorescent light coming from your sample. Pretty cool, huh? So now that we've got the science down, let's get practical. How does a lab actually do this stain? You're about to see that its beautiful simplicity is one of its biggest advantages. The whole process is refreshingly straightforward. You start by making and fixing your smear, just like you would for any other stain. Then you just flood the slide with the Acridine Orange solution and let it sit for about 2 minutes. That's it.
No complex steps or multiple chemicals. After that, a quick gentle rinse with top water, let it air dry, and you're good to go. The whole thing, from the raw sample to looking at it under the scope, can take less than 10 minutes. That is a massive win when a doctor is anxiously waiting for a result. And this technique is just incredibly versatile. You can use it to quickly spot malaria parasites in blood or to screen blood cultures to see if they're positive. In a scary situation like suspected meningitis, it can rapidly find bacteria in CSF, which should be totally sterile. It's even used for things like finding Giardia in stool samples. This flexibility means that no matter where you are, an ER, a public health clinic, a huge reference lab, Acridine Orange could be a key tool in your kit. So with all that versatility, where does this technique really make the biggest impact in the clinic? Let's look at a few key applications. This table right here is pure gold for your exams. It really puts Acridine Orange in perspective. Just look at its key advantage: speed. A Leishman stain is way better for figuring out the exact species of malaria, and a Zeal-Nielsen stain is the gold standard for TB, but neither can touch the incredible speed of AO for just screening. It's often used as that first-pass test to get a quick yes or no answer before you commit to the slower, more detailed tests. One of its most important uses, without a doubt, is the rapid diagnosis of malaria. I mean, think about it. In places where malaria is common, labs can be overwhelmed with hundreds of slides a day. Trying to go through all of those with traditional methods is a massive challenge. AO is a game changer here. The malaria parasites glow like little light bulbs, while the red blood cells, which don't have a nucleus, stay dark. It makes spotting the parasites incredibly easy. And the numbers really speak for themselves. In one study that compared AO to the gold standard Leishman stain, it had a sensitivity of, get this, 99.28%. That's incredible. It means if a patient actually has malaria, Acridine Orange is almost certain to find it. You're not going to miss many cases, which is crucial. Its specificity was also fantastic, coming in at 97.19%. So if a patient doesn't have malaria, the test is very likely to be negative. You're not going to be raising a lot of false alarms. This combo of high sensitivity and high specificity makes it a super reliable and powerful screening tool, especially in busy clinics where resources might be limited. But, of course, no test is perfect. It's really important that you understand both the good and the bad to know when to use it. So let's talk about the trade-offs. So yeah, let's weigh this out. The pros are pretty obvious. It's blazing fast, it's super sensitive, and it's easy to do. But the cons are just as important. The number one hurdle is you need that expensive fluorescence microscope. Plus, it's nonspecific. It'll tell you that bacteria are there, but it can't tell you if they're gram-positive or gram-negative. And there are some practical headaches, too. The glow fades, so you can't save the slides. And sometimes other junk on the slide, like cellular debris, can also glow, so it takes a trained eye to read it right. And we absolutely have to talk about safety. This is a huge point for your practicals and for your entire career. Because Acridine Orange works by literally squeezing itself into DNA, it's considered a potential mutagen. That means it's suspected of causing genetic defects. So you must, and I mean must, always handle it with proper personal protective equipment. Gloves, lab coat, eye protection, no exceptions. All right, time to wrap this all up. Let's boil everything down into those key, high-yield points that you absolutely need to know for your exams, for your vivas, and for those competitive entrance exams like NEET PG. Seriously? If you're going to screenshot one slide from this whole presentation, make it this one. If you can rattle off these 7 points, you've mastered Acridine Orange. What's the principle? Intercalation. DNA color? Green. RNA color? Orange. Why are bacteria orange? High RNA content. Main advantage? Speed. Main disadvantage? You need that fancy microscope. And the key safety risk? It's a mutagen. Got it? Good. And this brings us to the final and maybe the most important idea: clinical judgment. See, choosing the right test isn't just about what's technically the best. It's about what's best for your patient right here, right now. In an emergency, like if you suspect meningitis and need to know immediately if there are bacteria in the spinal fluid, the speed of Acridine Orange could be a lifesaver. You're making a conscious choice to trade the detailed info from a gram stain for the immediate actionable answer from a rapid screen. And understanding that trade-off, well, that's what's going to make you a great doctor.